Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. And right away, a big thank you to you, the listener. If you are finding these deep dives valuable, please take a moment to like, subscribe, and share this with your friends and family. It truly supports the Science Experience and helps us keep digging into this cutting-edge research. Today, we are undertaking a deep dive into an animal that truly defies biological odds. The bowhead whale, Balena mysticetus.
Oh, yes, the bowhead.
This creature is just a masterclass in, well, biological resilience, maybe not immortality, but living over 2 centuries while somehow maintaining near-perfect health.
And crucially, virtually immune to the scourge of cancer, which is kind of the main puzzle here.
Exactly. So our objective for this deep dive is to explore the brilliant detective work of researchers who zeroed in on the cellular and molecular strategies this massive mammal uses to achieve such extraordinary longevity.
Right. We're looking for the evolutionary secrets, the tricks that allow the bowhead to maintain its genome integrity over a time span, well, no other mammal can even come close to matching.
Let's just take a second to appreciate the scale here. The bowhead whale is monumental.
Oh, huge. Second largest animal on Earth.
Weighing in at over 80,000 kilograms. And its age isn't just guesswork, right? It's confirmed by rock-solid science like aspartic acid racemization analysis.
That's the one. Shows confirmed lifespans exceeding 200 years.
2 centuries. It's living roughly 3 human lifetimes. It's incredible.
And this scale, both in sheer size and time, immediately creates a fundamental biological puzzle. Peto's paradox.
Okay, Peto's paradox. The paradox being bigger animals with longer lives should get much, much more cancer. Is that the gist?
Precisely, yeah. Cancer is fundamentally a disease of uncontrolled mutation, of cell division gone wrong.
So the more cells you have and the longer those cells are around, dividing, existing,
the more opportunities there are for mutations to build up, accumulate, and eventually cause tumors. A bowhead whale has an astronomical number of cells.
Trillions upon trillions more than us.
And lives over 20 times longer than a human, on average. Statistically, if you just run the numbers, every single bowhead whale should be guaranteed to be riddled with tumors by the time it turns, say, 30.
But they're not even close. They are remarkably resistant to cancer.
It is one of the most profound incongruities in biology, really.
Yeah.
And it suggests their defense system must be completely unique, or at least highly specialized.
So it leads us to ask, what is the evolutionary choice they made? Because when scientists look at other large, long-lived animals like elephants, we see a kind of standard aggressive solution.
Yeah, we've touched on this before. Elephants have extra copies of key tumor suppressor genes.
Like TP53, that famous one. They serve as a sort of molecular kill switch, right?
Exactly. If an elephant cell sustains significant DNA damage, the cell doesn't mess around. It triggers hyper-enhanced apoptosis. That's programmed cell death, to instantly eliminate the potentially dangerous cell.
Get rid of the problem before it starts.
That's the classic aggressive strategy, elimination. Kill it quick. But the sources we looked at reveal the bowhead whale took a completely different path.
Okay, how so?
Well, their cells actually displayed lower basal activity of that crucial P53 gene compared to human cells.
Lower, not higher.
Lower. And maybe even more telling, after being stressed with DNA-damaging agents, the whale cells showed no significant increase in apoptosis compared to human cells under the same stress.
Wait a minute, that's the first major shocker here. The animal that lives 200 years and basically laughs at Peto's paradox has seemingly opted out of the textbook defense mechanism.
It seems so. It's not proactively killing off its damaged cells nearly as much as we'd expect, certainly not like the elephant.
Wow, so it's the opposite of the elephant's approach.
In that respect, yes. And this non-reliance on apoptosis suggests that the whale is not betting on aggression, on elimination. It's betting on something else.
Yeah.
Perhaps perfect upstream maintenance.
Okay, but that leads us to another really counterintuitive finding you mentioned, something that initially confused the researchers.
Right. The finding about how easy it is, relatively speaking, to turn a whale cell cancerous in the lab. This is where it gets really interesting.
Easier? How can that be?
Yeah. When researchers tried to force whale fibroblasts, these are connective tissue cells, into malignant transformation in a petri dish, basically, trying to make them cancerous.
Okay.
They found that the bowhead cells actually required fewer fundamental genetic errors, fewer oncogenic hits to turn fully cancerous than human cells did.
Hold on, fewer hits. Human cells typically need, what, 4 or 5 specific genetic failures to break free and start growing uncontrollably?
That's the usual number, yeah, 4 or 5 hits. But the whale cells only needed 3 in this setup, H8 HAIR, HRES, G12V, and the SV40 LTST equivalent.
Okay, I have to push back on this a bit. If it's easier to make their cells cancerous in a dish, doesn't that completely undermine the 200-year lifespan and cancer resistance? Why would evolution select for a system that's inherently, well, easier to break in that way?
That's the critical question, isn't it?
Yeah.
And it really flips our traditional thinking about defenses upside down. It suggests that maybe the whale has a slight vulnerability in those late-stage checkpoints, the systems that are supposed to trigger death or stop growth when things go really wrong.
Yeah.
But it compensates, massively compensates, for this potential downstream vulnerability by having an absolutely unparalleled system of upstream error prevention and correction.
Ah, I see. So the whale might have slightly less robust final safety nets, but it's traded that for hyper-efficient mechanics working constantly at the front line.
Exactly. They're just so good at fixing the damage immediately, right when it happens, that the damage rarely accumulates to the point where those late-stage checkpoints even become relevant.
So the whale cell isn't primarily designed to kill itself if it's broken. It's designed never to be broken in the first place.
You're right.
Or, if it is, to fix it flawlessly.
That nails it. This strategy of maintaining genome integrity through enhanced DNA repair seems to be the core defense mechanism.
Let's unpack the specifics of that repair then, because this is where the real magic seems to be happening. What makes their DNA repair mechanisms so superior?
Well, several things. First, their cells exhibit significantly lower mutation rates overall, both spontaneous mutations that just happen and mutations induced by external damage.
Fewer mistakes happening, period.
Right. And perhaps most tellingly, they are profoundly resistant to accumulating large structural variants. These are the big rearrangements, huge deletions, insertions, or duplications of DNA, especially ones over 500,000 base pairs long.
So they're preventing the really catastrophic DNA changes.
It seems so. The cells are simply making fewer errors, and the errors they do make are fixed with astonishing accuracy and efficiency.
And that accuracy is most critical when dealing with the really nasty stuff, like DNA double-strand breaks, DSBs. Those are the worst, right?
They are incredibly dangerous, yes.
Wow.
Like cutting both strands of the DNA helix completely. And the bowhead whale's DSB repair capacity and fidelity are significantly enhanced. We're talking about the two major repair pathways here.
Non-homologous end joining, NHEJ.
And homologous recombination, or HR, both showed higher frequency and efficiency in whale cells compared to the other mammals tested, like humans, mice, and cows.
Can you help us visualize the difference? If I break my DNA, how do NHEJ and HR typically fix it, and how does the whale do it better?
Okay, think of NHEJ as the quick and dirty fix. It's fast, it works throughout the cell cycle, but it's often messy. It basically just jams the two broken ends back together.
Which can cause small insertions or deletions, right? Like losing a few letters.
Exactly. Indels. It's error-prone.
Yeah.
HR, on the other hand, is the slow, meticulous restoration process. It uses the undamaged sister chromatid, the identical copy of the chromosome, as a perfect template.
So HR is accurate but complex and only works after DNA replication.
Precisely. Now, the bowhead whale, it seems to have upgraded both systems.
Okay, so if NHEJ is usually like duct tape, what's the bowhead whale's version?
It's more like precision-engineered superglue applied by a nanobot. The sources show that the bowhead whale's NHEJ was significantly more accurate, producing fewer deletions compared to human cells doing NHEJ.
Wow. They made the fast, messy system precise.
They improved its fidelity significantly. And it's not just DSB repair. Their mismatch repair, MMR, which catches small errors right after DNA copying, was also significantly more efficient than in human, mouse, and cow cells.
This really underscores that conservative strategy idea. It's not just about having more repair capacity. It's about the fidelity, the accuracy of the repair.
Yes. They seem to spend their evolutionary capital ensuring the cells are perfectly repaired and retained, allowing them to stay healthy and functional for, well, 2 centuries.
It's incredible. And that leads us to the identity of the master mechanic, maybe. The protein the researchers identified as potentially being a key molecular lever in all this.
Right. The protein CRBP.
Cold-inducible RNA-binding protein. Okay, tell us why this specific protein is so critical here.
Well, the first clue was simply its abundance. CIRBP was found to be markedly almost shockingly, abundant in bowhead whale fibroblasts and tissues,
Abundant compared to what?
compared to pretty much all other mammals studied. In most, it's largely undetectable or present at very low levels. You only see moderate levels in some evolutionary neighbors like dolphins and humpback whales, also long-lived cetaceans, notably.
Okay, so this high abundance looks like a specific adaptation and evolved strategy, particularly strong in these massive, long-lived baleen whales.
Absolutely. And the whale version itself, BWCIRBP, shows signs of evolutionary fine-tuning. It has specific amino acid differences compared to the human version, and its gene shows a higher codon adaptation index.
Which means
That's a molecular signature suggesting its messenger RNA is highly optimized for maximum translation, basically, for making as much of the protein as possible very efficiently. Evolution seems to have said, we need a lot of this specific protein, and we need it made reliably.
okay, so it's super abundant, and evolution has tweaked it.
Right.
What happened when the scientists tested what this highly abundant whale CIBP actually does?
The results were pretty definitive. When they took the whale's CIRBP gene and overexpressed it, made lots of the protein in human cells.
They put the whale protein into human cells.
Yeah. And it acted like a universal DNA repair booster. It dramatically increased the frequency and efficiency of both NHEJ and HR repair mechanisms in those human cells.
Wow. So it upgraded both the quick and dirty and the high-fidelity pathways.
It did. And importantly, it also reduced the mutagenic outcomes, those little deletions and chromosomal problems that usually result from incomplete or messy repair. It improved the quality of the repair.
So it increases quantity and quality. How does it actually do that? Do we know the mechanism?
The in vitro studies experiments done in test tubes helped figure that out. CRBP seems to act as a structural helper at the site of the break. It literally protects the broken DNA ends from being chewed back by enzymes called exonucleases.
Like putting caps on the broken ends?
Sort of, yeah.
Yeah.
And it also helps recruit and stabilize key repair proteins, like the Q70Q80 complex, which is essential for NHE, right at the damaged site. It's like a foreman concentrating the repair crew and tools exactly where they're needed.
It's coordinating the perfect fix. Okay, that makes sense. And they didn't just show this in cells in a dish. They demonstrated its effect on longevity in a whole organism too, didn't they, using fruit flies?
They did, yeah. The Drosophila experiment provided that crucial link between CRBP and lifespan. They engineered flies where they could turn on the expression of either human CRBP or whale CRBP.
And what happened?
Conditional overexpression turning it on in adult flies of both human and whale CRBP successfully extended their adult lifespan.
It made the flies live longer.
Yes. And maybe just as importantly, it also made them far more resilient. It significantly improved their survival after being zapped with lethal doses of X-ray irradiation.
Which is basically a direct test of resistance to catastrophic DNA damage.
Exactly. So this single protein, CIRBP, when highly expressed and evolutionarily tuned like in the bowhead, seems capable of improving multiple facets of DNA repair, boosting resilience, and extending lifespan. It's quite an elegant solution to the problem of aging and damage accumulation.
It really is. And this has potentially enormous clinical relevance, doesn't it?
It really does. For decades, I think the biomedical community largely operated under the assumption that fundamental DNA repair capacity couldn't be significantly or safely improved in adult mammals. We focused on things like apoptosis elimination, not enhancement of repair.
And the bowhead whale provides pretty definitive evolutionary evidence that this assumption might be incorrect, or at least incomplete.
Exactly. This creature, by apparently focusing its evolutionary efforts on preservation and precision repair, shows that boosting repair capacity is a viable, successful longevity strategy in mammals.
So the idea would be that maybe therapies based on increasing the activity or abundance of proteins like CIRBP could one day help.
That's the hope. It opens a completely new therapeutic avenue. Instead of just killing damaged cells, maybe we could help cells fix themselves better. This could potentially target the root cause of genome instability, which we know is a major driver of both aging and cancer.
And perhaps do it without the systemic toxicity that often comes with those aggressive cell-killing therapies like chemo.
That would be the ideal, yes. Targeting the repair process itself.
What stands out to me the most, just reflecting on this whole deep dive, is that ultimate evolutionary strategic choice. It's fascinating. The longest-lived mammal on Earth, the one that basically solves Peto's paradox for over 200 years. It doesn't do it by evolving better ways to kill its damaged cells, like the elephant.
No, it seems not.
Instead, it evolves these hyper-tuned molecular tools, like CRBP, to just perfectly repair them. It's a phenomenal example of precision beating aggression in the biological long game.
It truly is a remarkable illustration of how complex life can find ingenious ways to circumvent what seem like fundamental biological trade-offs, all through targeted molecular evolution.
Thank you for joining us on this deep dive into the extraordinary genome of the bowhead whale and the surprising power of proteins like CRBP. Don't forget to like, subscribe, and share this deep dive with your friends and family to support the SANS Science Experience. We'll be back soon with more of the latest and most exciting papers and preprints in science. Until then, keep digging.